My talk is somewhat related to the topics that David talked about, with a few marked exceptions. We work on entirely different length scale, different application, and the last thing we want is actually charge transport, especially not electrons. So let me start first by really explaining the application and the framework that we work with. And even though the primary characterization of the devices that we work with is electrical or electrochemical, we still rely very heavily on the biological interfacing. So I will spend some time describing that, because this is really the application, but also a very important element in characterizing how the devices are working, what the physics of these devices. So the application, generally, neurostimulation is a very wide field, and probably the holy grail in this field is electrical stimulation of cells, neuronal cells in the retina. We have about 1 million nerve cells in the retina that are each has an axon, a wire, that all bundle together and go through the optic nerve to our brain. And the information that the optical information that we receive comes from the rods and cones, and there's some internal processing that takes place. And one of the big challenges actually facing humanity, you may know or not know, that most of us, primarily Caucasians, have an astounding statistics of about 20% over the age of 80 to begin developing age-related degeneration of this layer. So this is actually, in terms of human evolution, even human society, this is going to be a really, really a problem where you basically become blind just because we live longer than this system can still operate. But leaving that bad news aside, this is a cross-section histology which shows those different layers. So David was talking about how limited cartoons are. So this is the cartoon, this is real life, at least if you're a rat. But it's not very different. So you have these cells here. These are the ganglion cells that are actually connected to the brain. And these are the two inner layers. If you cause degeneration, either deliberately or indeliberately, you can, what you see is this layer just vanishes. This is just, here they applied very strong light. So you can do that, but again, if you just wait long enough, most of us at some point would begin to suffer from this loss. The meaning is that even though we have the entire system still connected to the brain, there's no optical information transduction. So when light comes in, this layer is gone. And this opportunity has been realized long ago, maybe going back at least 50 years, that you can use electrical stimulation. Even though this process here is biochemical in nature, you can bypass this whole system and just place electrodes either here or here and apply electric field, and you would see. And this is actually the very first test that people did, was to use human volunteers who were waiting for an eye enucleation procedure because of cancer. You put an electrode in, you apply electric field, and you would see a phosphene. It's the sensation of light. Volta, by the way, we were reminded about some historical facts this morning. So Volta, who was the first person to build a battery, was the first person to put an electrode into his ear and to convince himself that by just applying, again, a very crude electric field, you can hear. So it's a long distance between an actual functional device, and this is the point. The point is that if we have 1 million cells and we're replacing them with 60 or 100 or 200 or 1,000 electrodes, this is still a very major problem if we think about vision restoration, visual restoration. And for AMD and other diseases that affect cause this retina degeneration, it's really the high resolution that is needed. So again, electrical stimulation of nervous cells is pretty easy, high resolution, at a very, very gentle conditions for long term is something entirely different. Now, I don't have the time, of course, to go through all the details, but just to equip you with the most important elements. So if we have neurons, neurons basically have voltage-sensitive proteins in their membranes, located usually at very specific regions, and this is what we're trying to target when we do electrical stimulation. So when we charge the electrode, we want to build up strong enough, not really voltage, but voltage drop over the cell membrane. So this is the important thing. We need about 10, 15 millivolts at the cell membrane, wherever the cell is located. So this is really important because it's not just about building very high voltage at the electrode, but it's actually building a very high potential drop of around 10, 15 millivolts where the cell membrane is located, hopefully where those voltage-sensitive proteins are. From the electrochemical point of view, again, this is where things become very, very subtle. It has to be done at high resolution. It has to be done in a way that we're not causing any Faradaic processes. So this is the main deviation from the world that David was talking about. We don't want any reactions to take place. So as I mentioned, people are talking about these applications for a long time. And so there are devices. There are actually people with such devices. Thing is that the aim is to reach a system that has no connecting wires and optimally will be able to convert light directly into electrical stimulation at the highest resolution and low power as possible. So this is some, to give you some more insight and just intuition into these processes. So what you see here are what we call microelectrode array experiments that we do with cultured cells. So these are cells from rats, neurons primarily, that are cultured on the surface. These are 10 micrometer electrodes, and every time you see these red marks, this indicates that we apply an electric field or electric current through the electrode, and you can appreciate the fact that cells are activated. So again, it's very, very straightforward to do it. What comes as a major challenge is how to do it in a wireless way. So it's directly optical to electrical stimulation. Here there's a massive rack with a current generator that is driving the pulse. And in principle, what we would like to do is to really understand what we're doing. Because what is very important to appreciate is that these processes can happen, David was mentioning again and again, artifacts. Anything can cause neurons to fire. If you heat up the surface, they would fire. If you knock on the table, they would, you could cause them to fire. So it's very, very important not just to make them active, but really understand why. So we have to exclude Faradaic processes, mechanical processes, and of course thermal processes. And we really want to validate that we're in this regime. And we want to be able to do that while also achieving high resolution and ultimately electrodes that we can efficiently couple to the system, and as you will see it towards the end, also in a mechanical biological manner. So again, some more insight about the bad things we can do. So here you can see the images you've seen before. This is calcium imaging. So we have a calcium sensitive dye, and we can mark each neuron, and we can see its activation. And you can see here an example where we took a couple of cells and stimulated them. And you can see that some of the cells at some point stopped responding. And the reason why they stopped responding is that we electroporate them. So this is another thing you can do. You can cause them to fire, but you could also damage them permanently or in a transient way. So the way, the traditional way of avoiding that is, of course, to go below limits that are causing thermal effects, electrochemical effects, and electroporation effects that people don't usually consider. So we have these general rules. Here again, another subtlety of this system. As I mentioned, the cells have their cell body with the high concentration of the voltage-sensitive proteins in certain region, but then they also have the axons, the wires that take the information to the brain, and these axons are sometimes as sensitive as the cell itself. So when you stimulate neurons, typically people think about stimulating the cell body, but this is not really the case. Very often, and this is what you see here, are examples where we go up and up in intensity, but we don't increase necessarily the diameter of the cells that we're actually activating, because we may already active cells that are very, very far away, just because we're activating those processes that cross the electrode. So this is another a really nice thing that you can do with this setup is you can run the electrical stimulation and not just to watch. I mean, here what we did, we looked at which cells, the statistics of response of different cells around an electrode. Here what we did, we picked one neuron and we marked all the electrodes that were causing that neuron to activate. And by doing that, we are basically mapping the areas where this cell is very sensitive. And you can see that this is really the classical picture. We have one neuron, one electrode, one neuron, one electrode. So we're really targeting those very high sensitivity regions of the cells. But these are two examples where we have an entire branch of processes, those axons and dendrites, that each one of them this entire region can activate eventually that cell to fire. The meaning of this is that this is eventually, when we try to think about vision restoration, this would translate to skewing the image that we think the brain needs to receive. And this is very, very important. And probably one of the last things that I want to tell you about the biological response or the tissue response, and this is a very important thing, is that now this is an experiment we did exactly with the same setup. So it's the same electrode arrays, but now instead of using cultured neurons, we use an entire cheek retina. So the retina can be removed entirely and placed across against the electrodes, and then we can stimulate at a specific region and then record the electrical activity away from the electrode. And the nice thing that we see with the specific model that we're using, which is the cheek retina, cheek embryo retina, is the time latency. So you see that as we go further away from the stimulating site, we have longer and longer latency. And this is really, you can really calculate the propagation velocity of the signal and compare that with what is known in the literature, and that makes perfect sense. So this really shows that what we're stimulating here can in some cases be the axons of the retina. Now, just keep in mind the structure of the retina and the fact that this kind of information is very, very important when we try to analyze what we obtain when we do the electrical stimulation. So this is the classical picture. This is the relatively short latency, okay, that we see here. This is 2 milliseconds after the stimulation, around 2 milliseconds. And this is the long latency. So this is after, we call this the direct response. So this is the response that is directly takes place after we do the stimulation. So the cells are firing in a pretty coherent manner. And this is the response that also involves synaptic activity. But, for those of you who are not familiar, this is probably too much. Okay, so what we can do and what people in this field have been doing is using electrodes that can be microfabricated or nanofabricated and can be eventually introduced to neuronal tissue, including to the eye. What people started thinking at some point was to use photosensitive systems. So silicon was a very obvious choice because silicon is a very good material for converting light and turning that into an electrical pulse. Now, silicon has been used, and there are a couple of devices based on silicon. The thing is that silicon is not a very good material in these settings for many, many reasons. Probably the most important one, which people don't often appreciate, is that in wet conditions, it's extremely unstable. It's just, I never could understand how silicon nanowires devices can even work. These things just come off at absolutely no time. So this is, silicon devices must be passivated, which really makes, it's counterintuitive. So the alternative came about when people started thinking about new materials, new systems that would be stable in wet conditions and can do this trick of converting optical signal into electrical signals. Probably the first systems that people were suggesting were P3HT, and there were various other systems. Now it's become more and more obvious that these systems do not really support direct electrical stimulation, but actually thermal stimulation. Since we're not material people, we're really focused on the applications. We tried these different systems with different colleagues, and the most successful system that we started working with a few years back is the system that was developed first in Linz and now in Linköping by Eric Glovatsky, and that's a pigment-based PN junctions that are fantastically stable in wet conditions, completely unpassivated. I'll show you some more of those features later. And again, unlike the framework that David was describing, the framework that we work with has is not dry, it's wet, and the solution is actually part of the system and a very, very important part of the system. So I tried here to kind of give you an insight of this new concept that we introduced, which is a semiconducting photocap. So what is a photocap? A photocap, this is the new device that we're using in these applications, is a capacitor that we activate optically. So this is the photodiode. It's a PN junction made of these pigments. And this PN junction is located on an electrode. Now, this is a very important part of the device because this electrode actually closes the loop with the solution. So we actually used gold, which is a Faradaic material, but you can in principle use titanium nitride or other non-Faradaic materials. Then you have the solution, which includes, so here basically you have solution and ionic conduction. Here you have pretty much standard semiconductor electronic conduction. And here the most horrible challenge, which is the interface. So running experiment with dry conditions can give you insight on improving this element. Otherwise, you really have to work with this system at its entirety. And as you would see, it's very sensitive to any parameter that we can introduce. So the theoretical or the modeling framework that we work with is very, very loose one. But it's because even modeling this diode, the diode doesn't have two contacts. It doesn't have two metallic contacts. It has one metallic contact, but then the upper contact is really the solution. So it's a very crude model. So this is the semiconducting pigments. They're really, really, really stable. I mean, working in the lab with these materials is really nice because you can go back to the same devices months back and they're still functional. So it's really going back to the days of semiconductors.
The effect that this can work depends on the carrier density in the solution.
Yeah, right. So what you're trying to do is to build here, say, few micrometers away from the electrode that 10 millivolt voltage drop. This is what you're trying to do without Faradaic processes. And so luckily, the group in Linz has been optimizing these materials and the fabrication of these materials for a few years, and the system was pretty mature when we started working with. And our task was really to characterize it and to demonstrate that it can actually perform this cell activation. So again, because we don't have two electrodes, and since we can't work in dry conditions, the system can be only characterized in wet conditions in various configurations that the most important one is probably this one, where we have the pigment films deposited thermally over a gold electrode. We have another electrode in the solution, and we can characterize the voltage. So if you look here at the VT, that would be the voltage that we measure typically 10 micrometers above the electrodes, just as a crude number that represents the location of the electrodes. And again, we don't need that voltage there. We actually need the voltage drop. And so you can apply different light intensities. So these are different light intensities, and these are the voltages that you build over the electrode. This, for example, shows you the difference between that exact light response as a function of the light intensity. So of course it goes up with light intensity. But it also depends on the solution, because again, the solution is the upper electrode. So the PBS is the conventional solution used in these applications, but ACSF models much better the conditions in the eye, and as you can see, that by itself has an effect. Then came one of the most important realizations that we made is the fact that I mentioned before, is that the gold electrode or the underlying electrode is playing a very, very important role in closing this circuit, right? I mean, we have this circuit here. So this is basically this film here. So here, again, it's modeled crudely as a double layer capacitor with a parallel resistor representing gold. Without this material, if the film is covering the entire surface here, this one here would be a very large load, and the numbers that we obtain, the voltages that we can build, diminish very, very quickly. And this is again something that we were a bit puzzled when we, well, before we were puzzled, we actually were very happy that we could, in this system, control this layer very systematically. But this is something that has been quite neglected when we compare with what other people are doing. Yeah.
Did you say how thick your layers were?
No, I didn't. It's a few tens of nanometers.
Few tens of nanometers?
Yeah. I'll mention it later. So the PN junction is a very thin LED system, and otherwise none of the other thicknesses really matter. So this is a very important thing, that if we don't have that layer around the pigment, that immediately reduces the efficiency of these systems. And so this can be seen here. We have the same diameter of the pigment pixel, if you want, and then around it we change the size of the electrode film, and this is what you see here. So the bigger the gold electrode, the smaller the load of the system, and the higher, dramatically higher voltages that you can build. This is, we call it infinity. This is just a uniform film of gold. And so this is was a very important realization, but that also limits the resolution. That also implies that we need enough area for every pixel, both for the diameter of the pigment, which clearly is a very important one. So the bigger the pigment film, the more charge we can pump into this PN junction, which makes perfect sense. And again, it goes up with the intensity. But it's also very, very important to have large enough electrode around it. And otherwise, the important thing to understand, again, comparing with the conventional approach of having two electrodes and having a closed system, this is done in a system where it's entirely open. So there is, of course, an equivalent closed circuit, but we're not part of it. I mean, we don't have access to electrodes when we characterize the system. And eventually, what we can do is we can, so all these were test samples. The samples here were realized on the same microelectrodes that I showed you at the beginning. So now we have, again, 60 microelectrodes. They are usually 30, here they are 30 micrometer in diameter. And next to them, we placed those new photocaps. So a disc of gold, and then at the center, a disc of the PN pigment junction. And so the first experiment we did, this is still a uniform culture, the calcium imaging that I showed you at the beginning. And what we do, we record the electrical activity. So this is not electrical recording. This is done with calcium imaging. We apply an electric field, and this is the control. So these are cells cultured directly on a bare material. Here we added the gold and the PN junction, and when we apply the electric field, we see the neuronal response. So this was a very nice initial demonstration, and then it was followed by basically the ultimate test. So now I'm pretty much using all the elements that I introduced to you so far. So these are the same results. If you look here, these are the same results that I showed you at the beginning. So we have a retina placed over these electrodes. So now we have stimulating electrodes, recording electrodes connected to a large external system. In addition, we have those pixels with the gold underlayer. And then we can either use external electric stimulation or photostimulation. So we can choose which region we're optically stimulating. The nice thing that you can see here is the alignment of the fibers. So you can really see the fibers. So they go along this direction. This is highlighted here. So we expect to see that latency effect that I mentioned at the beginning. So just to recap, we do electrical stimulation here at time 0. And then close to the electrode area, this is here, we see a response which takes place about 2-3 milliseconds later. And then if we go further away, we see that this latency goes a bit back, is longer, just because this is really the time that it takes the signal to propagate. So this is the conventional electrical stimulation. Now we do the opto-electrical stimulation. So we go to about the same region. We pick up that particular spot. We apply a flash of light at a wavelength that is supposed to be the most efficient. It's a short pulse of 2 milliseconds, very high intensity. And the nice thing is that we can reproduce exactly the same thing. Okay, so it's the same response. Now, if you, and this is a very, very strong validation that what we do is direct electrical response. Because if we were doing thermal process or electrochemical process, the response would not be the same. Okay, we've seen examples, and we can see from the work of some of the other players in this field that if it's not a direct response, if it's not an electrical effect, the effect would not be that short latency effect. It would be actually a very long latency effect. So this is a very, very strong further support to the fact that we see non-Faradaic, non-thermal process. Just to emphasize something that I neglected to emphasize before, generally when we look at the current response, when we measure directly the current going through the electrode, we see that we have something which is capacitive in nature. Okay, so this is not direct current. So if in this measurement you see something that looks like direct current, then you know for sure that this is not going to be capacitive process. The fact that we see here something which is capacitive-like doesn't necessarily imply that it's strictly capacitive, because it may still involve very strong Faradaic processes. So there's always still the ambiguity whether, in addition to the capacitive, non-Faradaic, the capacitive, the non-Faradaic processes we do, also do some Faradaic processes, and this is always a concern, especially when we do long-term activation with these systems. So this is really indeed the as far the most comprehensive analysis that we can do to really validate what we're doing. So in addition, the other important validation that you can do is really to go back to the model that I showed you at the beginning, the equivalent circuit, and go back to the literature and look at what typically people, what are the typical charge injection numbers that people use in order to do electrical stimulation. Because when you do electrical stimulation, the traditional electrical stimulation, you know exactly what current, what charge you are injecting into the electrodes. So this is a review from 2006, where they took different results from the literature. These are a group of physicists, so they plotted the threshold charge density as a function of electrode area, arguing that this is sort of the scale-free parameter that determines what is needed. And so you can say that roughly these are the values that are needed. There's still variation because these are many, many different systems, including in vivo experiments. But you know that from these kind of experiments, you know what roughly to expect. And this is a table of all the different experiments that we did over the years with different systems. So both with chick retina, with rat retinas, with cells from rats, cells from different systems, for different papers studying different things. And every time we get about, I really apologize for all these numbers. It's copy-pasting from Excel to PowerPoint. It came out pretty ridiculous, but the numbers are roughly around these values. So this is in millicoulomb per centimeter squared. This is the number. So this is around, here, which makes perfect sense. It's all consistent. It's not voodoo. We know what to expect, and trying to figure out from the measurements that we did here, we also get pretty much roughly the same values. And this is the sanity check, the ultimate sanity check, that we are using electrical stimulation. And again, the P3HT and actually quantum rods system that we developed along with Uri Banin a few years ago typically had much, much, much lower thresholds. So this really implies that it involves most likely different mechanisms. Of course, this still doesn't imply beyond any doubt that there are no other processes, but this is a pretty important sanity check. So I have 1 minute. So in 1 minute, I'll tell you, in half a minute, I will tell you what are still the challenges that we have. We take it very seriously to actually make this technology to work. Working means not just papers or patents, but actually, and not to blind animals, but actually to reach this stage of working with people. So we humans. So we're trying to develop a system. These are now the numbers. Sorry. We're trying to build a system that would be entirely implantable, built on flexible materials. It has to be really robust. This is a work that we've been doing for years now with a retina surgeon, trying to work out also how to glue the system. And in 20 seconds, this is an entirely different topic that we work on, but I just wanted to flash it to you because everything that we did, again, was involved in implantable devices, and implantable devices are ridiculously complicated because of all the things that I mentioned to you, but also because of the fact that regulatory concerns are really, really problematic. So a few years back, we started applying all this know-how that we obtained in developing different materials, different systems in electrophysiology, to actually develop wearable devices. So most of my group now is doing wearable devices, electrophysiology based on wearable devices. So this is recording electrophysiology, so exactly the same stuff that you've seen before, but now with people who walk about with entirely wireless system, and we can monitor, in this particular case, we're monitoring their facial expressions, and judging by your facial expressions, you need coffee. So I'll skip the all this. I'll definitely want to emphasize the people who did the work. So it was wonderful working with, and still we work with Eric Lovatsky and Serdar, who's just really fun people to work with. Most of the work that you've seen here was done by David. He was a postdoc and then a researcher, and now moved to the company that we established for the wearable devices. And that's it. So thank you very much, and I apologize for